I guess when did we first meet? 2003 at the math medicine thing I organized here, wasn't it? You gave a summer school and then talked about But we stretched back even further because
yes, actually, I remember because that was on the edge of when I could travel.
Yes, that's right. You were expecting a son,
a baby.
Right?
Yes, I was indeed.
And so then it turns out that we actually overlapped. But although I never met Helen at Oxford, I think I was probably starting my postdoctoral fellowship when you came in as a little baby PhD student, right? And I think Chris Budd was about a little bit junior to me, but about a contemporary because he babysat you for a little bit, right?
He babysat me.
Yeah. And then she was at Bath temporarily with my brother. She was she thought she'd escaped the civil O'Gonethans, you know, and she went to Manchester, University of Nottingham, and back at Oxford. And now there's no escape because we work in the same area. So anyway, it's a great pleasure to welcome you. This is the third in her distinguished lecture series. So
thank you, Siv. And I must say it's a real pleasure to be here, to have been here for the last couple of weeks. I am going home this afternoon. You will perhaps be sorry to hear. So this is the last of my 3 lectures, and with a sort of slightly populist title, Can Calculus Cure Cancer? And I guess perhaps preaching to the converted here today. And I guess before I start, and I guess Siv has already picked up on some of this, in some senses, I feel a bit like a cuckoo here today. And if I was here, maybe this is why I have to fly home today, in the sense that we haven't actually collaborated yet. We kind of seem to just keep like, I'm not sure if we're avoiding each other or what it is. But anyway, I hope that I can, at some stage before we stop working, that we can actually write a paper together. That is my goal. Anyway, so that's just by way of saying that it's really lovely to be part of this couple of days to celebrate all of the fantastic things that you have achieved over these years, but also to look to the future and say there's a lot more to do. And I hope that includes some collaboration. So what am I going to talk about today? The scene has already been set earlier this morning. So as we all are fully aware, radiotherapy is still a mainstay of cancer treatment. Yes, we can combine it with lots of other things, surgery, radio, chemo, all sorts of different other types of treatments. But nevertheless, most patients who have cancer will likely receive radiotherapy at some point in time. And I guess, again, as we all know, what does radiotherapy involve? It involves at some level directing ionizing radiation at cancer cells with the aim of either trying to kill them or at the very least to kind of control and limit their growth and spread. So that's great. And you think, well, what's the problem? And I guess there are lots of problems. Cancer is not a simple sort of object. Again, there are many different levels of complexity. And I guess this sort of diagram just really illustrates some of those challenges, which is kind of what makes it so difficult to know when radiotherapy given alone or in combination is or is not going to be successful. And I guess at the end of the day, what do the clinicians and or a patient want to know? If I'm going to give you a sort of rather unpleasant treatment, I want to know as quickly as I can whether it's doing you any good. And so I guess, as you can see here, some of the different levels of complexity, and I guess as we go through the talk, try to perhaps address some of these different challenges. Everything from, we have patient heterogeneity, no two patients, their life experiences, their biology, physiology are the same. That may well impact their response to radiotherapy. We hear a lot now about spatial heterogeneity, all the different subtypes of cells, whether that be different sort of clones of or lineages of cancer cells, vasculature, different types of immune and stromal cells, all contributing to the tumour microenvironment and influencing our response to radiotherapy. And I guess down, if we go further at the subcellular level, we heard already, I think this morning, about signalling pathways. We hear about epigenetics increasingly, and lots of detailed information that we can bring to bear. But nevertheless, these sort of just, I think, illustrate some of the challenges that, you know, when we're dealing with a real patient, that will influence their response to treatment. And so there are sort of three questions that I want to, three different studies to focus on today. Different pieces of work at, I guess, different spatial scales. The first is based on some work from a PhD student a few years ago, and this is really looking at how does treatment with radiotherapy impact interactions between different cell subtypes, and in particular, if we have a tumour that contains radiosensitive and radioresistant cells, how do they interact with each other, and does treatment with radiotherapy impact that interaction? Can we predict that? Then we'll move, and I think Kathleen did a lovely job earlier this morning about talking about clinical trials data from the Moffitt on head and neck. I'll pick up on that, but perhaps a slightly different take on how we can interpret clinical data involving patients being receiving standard of care radiotherapy. And then the final bit will be kind of going subcellular, and this is work that Julia did as part of her PhD. And there, what we're interested in is looking, I guess, at spatial let me say environmental heterogeneity. And in particular, if when we think about radiotherapy responsiveness, we know that oxygen levels play a role in that. If we find our tumour cells in environments where those oxygen levels are fluctuating, how does that impact their ability to progress around the cell cycle? And as a consequence downstream of that, how would it affect their radiotherapy response? So those are the sort of three sort of case studies that I want to share with you today. So first piece of work, as I said, this is really looking very much at how treatment, a single dose of radiotherapy, affects the interactions between two cell types, one which is putatively radiosensitive and one which is radioresistant. And this is work that was done a few years ago by a PhD student, Marcin Paksowski, I can't pronounce very well, Marcin. What I should say so is when Marcin came with a degree in computer science, and he was adamant that he wanted to do mathematical modelling and he wanted to do experiments. So he did the experiments and everything. So I really take my hat off to him. So what did we do here? So as I said, we had for two different types of cancer, PC3, these are, I think, both prostate cancer cell lines. We took different proportions of radiosensitive and radioresistant and co-cultured them together and grew up tumor spheroids. And this is just prior to any treatment, this is just the control. And then what we do is expose them to a single dose of radiotherapy and see what happens. But this here is just giving you an idea of how co-culturing two cell lines, how it affects their growth dynamics. So you can see in this particular case, we've either got the control radiosensitive or the resistant growing on their own. And then in this particular set of studies, we take a 50-50 mix, and you can see images of what those different spheroids look like for my PC3 and DU145s. And you can see the growth kinetics over on the right-hand side. And so the question that we wanted to ask here is, can we predict how these two cell types are interacting with each other? Are they antagonistic? Are they competing for space, resources, etc.? And do those interactions, or is the resistant one in some sense dominating the other population? What's going on? Can we work that out from these data? And furthermore, do those interactions change when we expose these cells to radiotherapy? And I guess here, what you can see is we've got the control spheroids shown at the top, and we've got the growth dynamics following exposure to single dose 6 grays of radiotherapy. And I guess what you can see is, let me just get my head around this. So on the top, you can see that the control and resistance, in the absence of treatment, their growth dynamics are different. And I guess they're still a bit different following radiotherapy. By contrast, for my DU145s, when I grow them sort of in isolation as a sort of homogeneous cluster, it's very difficult to detect any difference between the DU145's control and radio resistant. So this is kind of telling us from the get-go that different cell lines, different mutations are likely to have quite different sort of interactions potentially and or their growth dynamics. Yeah. So this is control, this is resistant, and this is no radiotherapy. Then I expose them to 6 grays of radiotherapy, and then I come back at day 3 and 6. And you can see there's not much of a difference. So just eyeballing this, you might say, are these controls really radio sensitive? Okay, but on the top, you can, I think, see a difference if you look at the sensitive ones. Okay, and so have I gone? No, I didn't have any growth curves. So simple model, just Lotka-Volterra. So if I just have monoculture, my tumor cells are undergoing logistic growth with a growth rate R and a carrying capacity. And then we have an interaction term, lambda R, which is the influence of my resistant on the radiosensitive or control, and similarly for lambda C. And if we just take the monoculture, then we can fit our logistic growth models with the lambda terms switched off because we've only got one homogeneous population, and we can estimate the growth rates and the carrying capacities for the 2 different cell lines. And you can see that the growth rates for the DU14, well, there's not much difference in the growth rates of the 2 cell types, but we predict sort of markedly different carrying capacities. So this is nothing terribly exciting. I guess the question now is, when we layer on top of this our co-culture experiments, then really what we're chasing here are these interaction terms. And I should say, when these experiments were being designed, I was very adamant that we needed a lot of data, because normally they would just measure at the beginning, maybe after a few hours, and then close to the end. And I felt that wasn't going to be enough. And so at some pain, the data was sampled much more often than they might otherwise have done, which I'm not sure how much they enjoyed. Because ultimately we knew we wanted to try and hunt down these interaction terms. But then when we started trying to fit the Lotka-Volterra to the model, we found we couldn't estimate the interaction terms with any degree of confidence at all. We just couldn't, we just couldn't do it. And then, so what we decided to do instead was to try to use our model to say, okay, if you just measure that more and more often, it's not helping any. Really, what I want is some information on the proportions. And they were quite annoyed when we told them this. And so what we decided to do, because they thought because in order to measure the proportion of control and resistant at each time point, they've got to disaggregate the spheroids. It's a pain. And so we tried to find a workable compromise, which was, do we really need to measure the proportions every single time point? And so we did some experimental design to work out how many times we needed to measure the proportions in order to be able to get a reasonable estimate of these interaction terms. And so with that sort of synthetic experiment, we predicted that 2 or 3 time points should be sufficient for us to be able to nail down these interaction terms. And then because they thought they were going to have to do it at every time point, then they were actually willing to do this. And so the result of that, so what we do is we've got our data, we take 50-50 or different proportions of control and resistant, and we grow them out over time, and then we can see what are the proportions of the two cell types at 3 time points through the growth trajectories. And then just with that layered on top of a more finely resolved data for our growth dynamics, what you can see here, I hope, the data still is a little bit noisy, but at the top, we're just fitting, I can't remember, I think it's whatever, PC3s, I think, one of the two cell lines. And I hope you can agree that we get a reasonable fit to both the control, no radiation, and the irradiated. And you can see, so the green lines are the extra data that we collected, telling us what the proportions were. And you can see that it's tracking, I think, quite nicely the growth dynamics, both of the control, no radiation. And I guess each of these different plots is giving you fits to the data for different initial conditions where we either have just control, 3-to-1, 50-50, or 1-to-3, etc. Different initial conditions in terms of ratios of the two cell populations, and likewise being able to predict the dynamics for the irradiated. And so with that extra data, then we were actually able to, we've already, if we want, estimated what the growth rate and carrying capacity for our two cell lines are in the absence of co-culturing them. With this extra data, then we can work out what the interaction terms were. And this is where it sort of became a bit interesting. So for one cell line, the PC3s, we found that the interaction was competitive. So both of those interaction positives were positive. So they're both competing for resources or space. But for the DU145s, we were quite surprised to find that it was an antagonistic relationship. And so the presence of the wild types was actually beneficial for my radio resistance. And this, I think, the experimentalists would never have thought that such an interaction could occur. So actually the modeling exercise was really insightful for them and stimulated them to go on and then look at actually the spatial distribution. And I think there's a lot more work that goes beyond this. And I hope it illustrates how even with some very simple models, how they can help with inform experimental design. And in this particular case, show up some interesting biology, which was not at all anticipated prior to doing this work. So I guess this just summarizes what I've just said. And I guess the other point to make is what we found was that the exposure to a single dose of radiotherapy, it didn't affect the nature of those interactions, but it altered the magnitude of those interaction terms. And so this was an original piece of work. And then, as I mentioned, a sort of interesting follow-on would be to try and understand why we're seeing these different sorts of interactions for two different cell lines. And then that begs the question of what's the spatial distribution of the cells. And so indeed, the sort of one of the experimentalists who is involved in the project, she went on and has done more work on spheroids where we can stain and see the relative positions of the radiosensitive and resistant, but we didn't have enough data and she went to Sweden, I'm sad to say. But in some follow-up work with Hayrim Cho, who was here last week, and a group of other early career researchers, we did some follow-up work where I guess the problem with the experimental data is we have no ground truth. We can just fit the model to the data. By using, so the advantage of this study here is we can generate, well, we can both generate perfect data from our Lotka Volterra, try and fit and see if we can come back and estimate the parameters correctly, do structural and practical identifiability analysis, look at different ways of how we fit the data. Do we pool the different experiments? Do we do it sequentially, et cetera, and that's all explained in the paper. But additionally, what we were interested in is trying to understand what spatial configurations might give rise to this antagonistic behavior that the model was predicting. And so we ran a bunch of agent-based simulations, very simple, where the interactions are either, competitive, mutualistic, antagonistic, generated synthetic data that mimics the experimental data, and then try and fit our Lotka-Volterra model to different agent-based model simulations. And I guess the question here was, when we've generated the synthetic data from an agent-based model with known parameter values, how well are we able to, what do those parameter estimates, how well are we able to infer them from the agent-based model data? And what this slide is really just showing you is for different contexts for our agent-based model, whether the interactions are competitive, mutualistic, antagonistic. If we vary the initial proportions of sensitive and radio-resistant cells at the when we seed our tumor spheroids, you can see how the parameter estimates that we get for those interaction parameters, that they are, some of them in particular, quite sensitive to the initial proportions of the, and I guess this in some ways illustrates the challenge of being able of estimating accurately model parameters from experiments. And I guess here, because you can see quite clearly, I mean, they're all generated with the same parameter values, but our predictions are quite different. And if you want to learn more, please have a look in that paper. I probably haven't done it justice. That really was just by way of a study of showing how we can apply mathematical modeling to in vitro experiments to try and understand something about not really the direct effect of radiotherapy in terms of cell kill, but what the longer term impact in terms of how the interactions between different cell types might be altered as a result of that exposure. What I'll move on to now is moving up a scale or two and looking at some clinical data from head and neck cancer patients from Moffitt. And this is work that was originally begun by a PhD student, Tom Lewin, and then finished in collaboration with Alex Browning, who's a postdoc in Oxford, but will soon be heading to a faculty position in Melbourne. So the data that we had here, and I think this is probably quite similar to what Kathleen was talking about earlier this morning. So we have head and neck cancer patients, CT scans, the lesions, which I'm glad I'm not a radiologist because I would find it difficult to find where the tumours are. They're apparently these blue things. And again, one of the nice things about this study is that the data is much more time resolved than would normally be the case for patients undergoing radiotherapy, where you might have a scan when you go to the clinic, a scan at the start of treatment and a scan at the end. Here we've got data coming in sort of, well, you can see 2 weeks, much more frequently. So this is, while the volumes might look a bit fuzzy, but the time resolution is really quite exquisite. And so then you would ask, what sort of mathematical model shall I fit to this time volume data? And again, Kathleen did the job for me very nicely early this morning. And one option would be to say, okay, between treatments, I do my logistic growth rate R, carrying capacity K, and I saturate to that carrying capacity. I then hit with my radiotherapy Monday through Friday. Each time I have an instantaneous cell kill where my tumor volume jumps by a fraction, which depends via linear quadratic, depends on my radiotherapy dose D. Alpha and beta are parameters depending on the particular cancer or patient-specific parameters. And so if we simulate, then the sort of behavior we might expect to see here is pre-treatment, my patient is trundling along on their logistic growth journey. I start treatment at day 0. And then we see this sort of classical sawtooth dynamics. I hit with radiotherapy, I see a jump in the tumor volume, tumor then regrows, I come back the following day, and so you see this zigzag. And I guess if treatment's working nicely, then we'll see a progressive reduction in the tumor volume. And you can see that sort of playing out for 2 different patients who've been engineered with different carrying capacities. And that's all sort of quite classical. And these would be growth dynamics that you would associate with a good response to treatment. And I guess a starting point would be take that data from Moffitt and fit such a model to it. However, this is not all of the data conforms to those dynamics. So we have one patient up here. So these are showing you the time courses measured at sort of whatever it is, t equals 14 days, 24, et cetera, So this one, if you imagine the sort of volumes on the intervening days, is sort of giving you an example of a patient who's responding nicely, but not everyone responds in the same way. So what you can see on the top right is someone who is really not responding at all. We can see a patient who responds initially and then they seem to plateau. Bottom right, we've got someone who seems to be not really responding terribly well initially, and then they start to respond. So if we use that simple model that I showed you on the previous slide, we're not really going to be able to capture all of these dynamics, and that's a little bit disappointing. So what we wanted to do is propose a new model, but we wanted to keep it as simple as possible with minimal addition of extra parameters in order to try and explain these different dynamic behaviors that we see within the patient cohort. And so the idea or the hypothesis that we had, and I'm not saying that this is the right answer, it's just a possible explanation, was to say that when we irradiate, implicit in the linear quadratic is that dead material just magically disappears straight away. So what if we assume that dead material degrades with a certain half-life? And so what we propose is a 2-compartment model where we have our live or viable cells V as we had before, but when they die, either through natural cell death or necrosis, or when we hit with radiotherapy, the dead material is held in a pool of necrotic or dead material, which decays with a certain half-life. Okay, so this is our sort of model 102, and then we try and fit this to the data. And indeed, the curves that I showed you, if you notice, the solid lines are our fits to the data, and the sort of shaded area around are our confidence intervals. So what we find is that with that simple extension to the model, we're able to reproduce all the different dynamics that we see in our patient data. Yeah, it's very, very simple. So we have our logistic growth. I've included a background rate of natural cell death. And then that when a cell dies by necrosis, that creates dead material. And then when I irradiate, so rather than, for simplicity, rather than having linear quadratic in here, I've just got a death rate gamma, which would be a function of my radiotherapy dose. So I'm just black boxing that into a single parameter. Does that make sense? And I hit with radiotherapy at discrete times, t equals 1, 2, 3, 4, 5. Yep. And then that dead material is moved into the dead compartment. And all of the dead material decays with a half-life zeta, if I can remember my Greek. And the idea of what might be, so it may just be natural degradation. It may also be infiltration by phagocytic immune cells. And so you could imagine that variation in that decay rate may tell us something about how competent my immune system is. Am I getting lots of phagocytes being recruited into the tumor to clear off that material, right? So there may be a number of reasons why that might vary from patient to patient. Okay, does that make sense now? Thank you. Yeah, please ask. I'd rather not finish, and if it makes more sense. So we fitted our model to all of the different patients. And surprisingly, what we found is, I know it looks a bit fuzzy, but the fitted parameter values seem to cluster based on a sort of qualitative notion of their response. And I guess let me in particular draw your eye to maybe the box that says, so we've got the parameter measuring radiotherapy cell kill, that's on a log scale. So many of these parameters are on a log scale, which is why they look sort of negative. It's a log of the parameter value. So radiotherapy cell kill parameter and the growth rate, if you look in that box there, you can see my fast responders have got, I guess, they're clustered with a certain growth rate, which isn't too big, and a certain radiotherapy cell kill. If I look at my poor responders relative to them, their prediction is a lower growth rate. So I'm going to be killing off fewer of the tumor cells. And I'm also, well, so a slower growth rate, but an even more marked reduction in the cell kill due to radiotherapy. So that's the fuzzy sort of orange halo that you can see in the bottom left corner. And you can see, for example, my pseudo progression, this magenta curve, we've got a very high growth rate, but we've also got a very rapid cell kill. So we're creating a lot of dead cells, and what we're probably doing is they're not degrading, which is why we see, so we're left with proliferating cells and this big mass of dead cells. The proliferating cells proliferate rapidly, and so that's why we see progression until we start to degrade the dead cells. Okay, so again, I was quite surprised that the parameters just do seem to cluster in some sort of reasonably interpretable manner. Okay, yeah. They're all on there. They're just, some of them are not so easy to see, especially on this board. They're all there in all of the different boxes. Oh. And I guess if you look at some of these, you can see, so again, without doing, you know, there are questions about how well you can identify all of these parameters. Some of these are quite fuzzy. But again, you've got like a bunch of different, you know, quite heterogeneous patients. So I think the fact that we're able to get any clustering out of this, I think, is quite, and it's not a very big study. We're talking about 40, 45 patients here, that's all, right? Yeah, okay. The other thing, of course, that we can do, because going back to what I said at the beginning about what does a clinician or a patient care about? If you're giving me a treatment that's not very nice and it's not doing me any good, I'd like to know that as soon as possible. And then we can stop and we can talk about doing something else. So the idea here was to try and use, so we can fit our model to a training data set, and we've got baseline levels then for, depending on whether I'm a pseudo progressor, a good responder, et cetera, and we can run our model with unseen data, and then we can try and predict the patient outcomes. And what you can see, very much as you might expect, so here are typical patients taken from each of those different groups, and if we just have the first 2 time points, unsurprisingly, very difficult to say anything very much. But you can see that as we successively add more data, then our certainty in being able to predict what the outcome is improving. And I guess furthermore, some of these different outcomes are seem to be easier to track than others. But again, I think it illustrates to you the benefits of having more time-resolved data, the actual clinical power that could deliver. Okay. So just to summarize what I've said there, it's a very simple model. So we look at logistic growth radiotherapy. That isn't going to enable us to understand all of the different qualitative behaviors we see in our patient cohort. We make a very simple modeling assumption that when we create dead cells, either a natural cell death or a radiation, that dead material isn't removed immediately. It has to be removed over a certain time scale. This new model is consistent with the data, and we find that the parameter values cluster reasonably well by patient outcome. Okay, so now let me tell you a secret. So I present that and it's all like wonderful and you think, oh, that's great. And she just worked it all out. Fantastic. Let me tell you how it really happened. So this was Tom's PhD. And what we were initially doing is looking at multi-phase models, which are quite a bit more complicated. And Heiko gave us this data and we were trying to explain it. So we developed this much more complex three-phase, multi-phase model where we distinguish tumor cells, dead cells, and extracellular fluid. If you're interested, there's a couple of references here. And, you know, you can write down mass and momentum balances. If you're interested, I thought I would spare you all of the equations, but they're quite complex, detailed PDE systems on with free boundaries where the domain size reflects how big your tumor is. And what we were interested in particular here is, because we kind of introduced this extra phase called dead cells, then there's a question of what sort of material properties do dead cells have? So we think of extracellular fluid as being sort of like an isotropic fluid. We think of our tumor cells as being some sort of viscous fluid. What's dead material? Because it's somewhere between those two. And so what we did was we had a parameter or tuning parameter that enabled us to dial between the two and to see what difference it would make to the sorts of predictions the model would generate.
At this stage, you don't care if the tumor is solid or
so I think what we're doing, you have to, I mean, so it might seem like a bit overly simplistic to say that a tumor cell is a fluid, but I think it depends on what timescale you're talking about. So if you're talking about on a very short timescale where you might probe it, and then it would have some sort of elastic recoil. But if you're talking about on a very long timescale where they're growing and they're moving, then in some sense they can be approximated by, say, a viscous fluid. Yeah. Yeah. So we view our tumor cells as sort of viscous-like on an appropriate time scale. And then we view our extracellular fluid as being isotropic fluid. And then we can think about the dead cells as being somewhere in between. Because in truth, your dead cells were, when they die, they're cells. And then as they degrade over time, they will kind of change their sort of material properties, if you like. And so it wasn't obvious to us what to do. And I should say this is just a purely qualitative study, because as you can imagine, just from the description, there's a heck of a lot more parameters going on in here, right? This was really just to see what would the qualitative behavior be in the absence of any radiotherapy, and then we layer on our radiotherapy on top. Okay, and if you want to see the details, then I'm just trying to not go too much into the weeds of the actual PDEs, because I thought you might not appreciate that too much. So really just to give you a sort of flavor of what the model predicts. So if you look on the plot on the left-hand side, this is that three-phase, much more sophisticated model showing its growth dynamics up to some time t equals 20. I give a single dose of radiotherapy, and then I chart its rebound following that exposure to radiotherapy. And the red dots are showing you the time points at which we look inside our tumor and we look at its spatial composition. So if you look, then the orangey or mustardy color at the top, that's telling you what proportion so on the x-axis, we have spatial position. So this is the outer tumor boundary. This is the tumor center. It's radially symmetric. So this is the proportion at a given spatial location that's dead material. This is the proportion that is, I'm going to get this right. This is fluid. This is dead material. And this is live cells, okay? So at the top at t equals 20, I'm having nutrients being supplied externally, they diffuse in. So you can see as you move towards the center, that yellow vertical line is telling you where the oxygen concentration falls below a hypoxic threshold where the cells stop proliferating. So you can see you've got predominantly proliferating cells, a little amount of cell death, and a certain amount of fluid. And then as you move towards the center and the cells are starved of oxygen, you see this central core full of dead material, right? And then I hit with radiotherapy, and then we look just after radiotherapy, and what we find is if you compare those two slides, radiotherapy preferentially will kill well oxygenated cells. So you'll see the dark blue has decreased dramatically, and it's been replaced by dead material. And what you'll notice is that yellow line, because if I kill off all of those cells, that will influence what the oxygen distribution is like, because there's not so many cells consuming oxygen. So there is no longer, all of the cells in my domain are well oxygenated. I think, oh, sorry, I tell you a lie. Sorry, I couldn't see it. The red line is the hypoxic threshold. The yellow one is the necrotic threshold. Sorry, I couldn't see. I was just trying to puzzle. So there's a red line, which isn't very clear to me here. So up to the red line, everything's well oxygenated. Red to yellow, I'm sort of hypoxic, I'm not proliferating. To the left of the yellow, I've not got enough oxygen, I'm just dying, right? So when I irradiate, I lose my necrotic center because the oxygen concentration shifts. More of the tumor is well oxygenated. And so all of the cells from here to here can now proliferate where they couldn't before. And then over time, as the tumor regrows, you'll see that the position of these interfaces is how it's changing. So I guess what you get for the extra complexity is it allows you to see inside the box and see how the spatial composition is changing over time, admittedly in a rather idealized sort of scenario.
Is it possible we might understand the dynamics of this interface?
Yeah, so there are several different interfaces here. So we have an outer tumor boundary here that's changing over time. We have two internal free boundaries, which are defined by contours of my oxygen concentration. And they will influence what my cells, their cell behavior. So where oxygen is reasonably high, then the cells will proliferate. That's to the right of the red vertical line. And so you have to track them implicitly. So in truth, there's sort of many more simpler versions of this model, interesting types of free boundary value problems. Yeah, lots. Yeah, so Avner Friedman is your man, I think, in terms of lots of interesting questions in proving existence, uniqueness, etc. For this class of problem. With the multi-phase, I think it's a little bit more challenging to do that, and particularly with a three-phase model, but certainly some of the simpler ones, yes. So that's just a single response. And then again, cut a long story short, this is just, so as I said, we did this work before we did the two compartment model. And so what we find is as we search through parameter space, but the parameter space we're searching through here is a much larger one, and it's much more difficult to know how to navigate it. But certainly what we did find is we were able to find parameter sets which reproduced each of the different qualitative behaviors that we saw in our clinical data. So here, these shaded lines is the Monday through Friday when I'm receiving radiotherapy. And you can see we've got our fast responder, et cetera, the same qualitative behaviors that we saw in the experimental data. So this is like an over-engineered model. It's very, very complicated. And when we could sort of look inside the box and see what was happening, then that motivated us to go back and say, we must be able to do this much simpler. And so then that was how we landed on the simpler ODE model, right? So, I mean, in a sense, and you can also do a lot of other sorts of experiments. I was mentioning before, what are the properties of that dead material? Does it matter? How should we estimate what that is? And this set of simulations here are really just giving you an idea of the impact that the material properties of that dead material have on the growth dynamics. So for the red line, I grow and then I irradiate. I'm treating my dead material the same as the extracellular fluid, isotropic fluid, and you can see we have elimination. By contrast, if I treat that dead material, that's the only difference, as viscous, more cell-like, then we see quite different growth dynamics. And indeed here we see that the tumour sort of evolves to some sort of equilibrium. Okay? So just really to make the point that it makes a difference to the predictions. So I guess to sort of summarize what we've shown thus far. So in truth, we started with a 3-phase model, tried to show that it could be consistent with our experimental data. Way too many parameters to have any hope in hell of fitting that to the data. So let's try and pull it back to something which is much simpler, and that's our 2-compartment model. I think there's a lot of interesting questions that this work, now I tell you the truth, that it throws up. And indeed, some of the things I would really love to do is to explore the relationship between the 2 models. Can we learn anything about the parameters in the complex model? How do they map onto the parameters in my simpler compartment model? And I guess just as an aside, we have extended these sorts of models. Here we had 3 phases representing live cells, dead cells, and extracellular fluid. There are many extensions to include other species, whether that be immune cells, blood vessels, etc., etc. So you can generalize these sorts of models to investigate more at a continuum level, more complex tumor structures. Okay, now it's 1:45. I've got one more thing to talk about. What do you think? How long have we got? Okay, we'll try, right. So final bit then is just going now to think about a sort of slight shift of gears, I suppose, but going, I guess, subcellular and thinking about how oxygen levels, which we know radiotherapy is sensitive to oxygen levels, but how do oxygen levels impact cell cycle progression? And I guess, and as I mentioned before, this is work by Julia. And as I've said, low oxygen levels we know are quite prevalent inside tumours. They disrupt the cell cycle, and as a result, that will likely impair my response to radiotherapy. But more than that, in certain types of tumours, we can also observe a phenomenon known as cycling hypoxia. And in certain types of tumours, you'll see spontaneously sort of regions of the tumour where oxygen levels just seem to fluctuate. And that might be over length scales, as you can see here in these mouse studies of, I don't know what that is, possibly not very much, maybe a few millimetres, and on time scales that range from seconds to minutes. I think that's about right. So what we're interested then in doing is seeing how do different oxygen tensions, how do different oxygen sort of protocols, fluctuating oxygen environments, how does that affect our cell cycle? And I guess we were fortunate here to have, again, experimental data, and our experimental collaborators have an oxygen hypoxia hood where they can take their cell lines and they can put them in oxygen normoxia for, say, 4 hours, then 4 hours of hypoxia, etc., etc. And they can do, at the end of the time, they'll take them out, put them through flow cytometer, and look at the proportions in the different phases of the cell cycle. And so we have, or Julia, we had data from different protocols. So if I just culture in normoxia, then after a certain period of time, I can see what the proportions in G1, G2S, or G2M in the first bar. And I guess really the take-home message from this is that the distribution of my cells around the cell cycle changes depending on the environment in which the cells are being cultured. And if we have a model of the cell cycle, how can we marry that with these different results? Kind of the question. So a classical model of the cell cycle might be just a series of compartment models. If you want to be a little bit more sophisticated, then you might have an ODE for cells in G1. Then that enters the DNA synthesis, where we have some sort of structured model where we keep track of the amount of DNA in the cell as it replicates its DNA. And then we go into G2M, where the cell finishes division. So we have ODEs counting the number of cells that are in G1 and G2M. And then we have a structured model which keeps track of how many cells are in my S phase, and in particular where they are in their sort of the DNA content that they have as they move through that compartment. Does that make sense? So we've got a schematic representation of the model on the right-hand side. Okay. Now, the problem with that model is if you want to now put different oxygen environments on, how do you do that? And in particular, though, that model won't be consistent with the data that I showed you if we start playing around with different environmental conditions. And so after, I think, quite a lot of pain, perhaps, exploration, maybe is another word, we came up with this extended version of that model. And let me just see if I can explain. So we have our DNA synthesis, that's in the structured place. We have G1 and G2 as before. And what we do is we subdivide this compartment. We say that under hypoxia, let me see if I get this the right way round, that the cells will stall, they'll go into a paused state, C1. And then when oxygen levels improve, they'll come back out and start cycling, right? So there's a pause state here under hypoxia. And then likewise, we have a C2, where again, cells will stall under hypoxia. But we know that hypoxia can induce damage to our cells. And so we have a damage state, D2. So when I come out of DNA synthesis, if I've experienced lots of low oxygen and I'm trundling through doing DNA synthesis, I need to check what I'm like, whether I'm competent to divide. And if I'm not, when I get out of the DNA synthesis phase, if I've got too much damage, then I'll just stop and die, okay? So we extend the model by adding in these various different compartments. And indeed, we can make up a number of different combinations where we retain or ignore different elements of the sort of full model that I just described to you. And you can see, so we have the full model with 7 parameters, et cetera, as we neglect different parts of that model that I've just described to you, sort of reducing, dialing down the complexity. Okay, and then what we can do is fit each of those models to our experimental data. And I'll show you the data, I think it's on the next slide. And what we can then, if we compare these models, penalizing for the numbers of parameters that they have, then we can calculate various different information criteria to see which of the models are most consistent with the data. And at the sort of bottom line was that it wasn't the most complex model, it was this intermediate model M1 that seemed to provide the best explanation, most consistent with the data. And sort of just to give you an idea of how well that model performs, so the data was pretty limited, and it's amazing that Julia was actually able to fit anything to it. And on the left-hand side, you can see the sort of data and the fit to just sustained chronic hypoxia. On the right-hand side, you can see the predicted response to cycling. I think it's 4 hours on, 4 hours off. Okay, and so you can see how the proportions in the different phases of the cell cycle evolves in these different oxygen environmental conditions. I guess you can do other things then. We can use the model to predict the response to environments that we haven't seen. And as you can see here, and as we change, let me see if we have, let me get my head around this, if we have a larger, if we have more rapid, say, fluctuations of cycling hypoxia compared with on the top panel, we've just got a single period of hypoxia. and then, no, wrong way round. So normoxia, then I switch them off, and then you can follow what happens to the cells over time. In this case, I expose them to fluctuating hypoxia over a period of time, and then we put them in hypoxia, and you can compare what the growth dynamics are afterwards. And in particular here, you can see how the exposure to fluctuating hypoxia, how that's impacting the number of damaged cells. It's having an impact on the sort of behavior of my cells when they come out. So it suggests to us that cycling hypoxia can increase the number of damaged cells. Damaged cells is equivalent to, I guess, increased potential for genetic instability, mutations, and bad things. So I guess just to complete this bit of work, I guess what it's trying to show you is there are many existing models of the cell cycle of varying degrees of complexity, but it wasn't clear, I think, because people haven't asked the question about how well do those models do if we want to consider what happens under fluctuating environmental conditions. And so what I think Julia very nicely and systematically did was try to propose a new model that allows us to kind of investigate the impact of exposure to different oxygen protocols on progress through the cell cycle. And I guess downstream of that, that puts us in a nice position then to look at, if we layer on top of this, cell kill due to radiotherapy. Again, you might ask the question, I sort of said there is cycling hypoxia, it happens in tumours. Another sort of piece of work that we're doing, but I don't have time to go through here, is to try and ask what is it that creates these regions of fluctuating oxygen levels. And so in separate work, we're looking at models of tumour blood flow through very simple motifs and trying to see if we can generate oscillatory blood flow that might explain the sorts of experimental observations that are seen in certain mouse tumours and also human tumours. So I think with that, that's probably enough from me. I've tried to, I think, tell you about 3 different pieces of work about how we can use mathematical modelling, and data, I think, is a common theme through each of these, to develop models of different levels of complexity to try to answer different sort of biological questions, to generate insight, to inform experimental design and other things. Acknowledgements. Martin, Hayram, Ali, Katie, Tom, Alex, Julia, I think, are the people who really did the work. Funders. Back to the question, can calculus cure cancer? Thank you.
Okay, thank you very much, Helen. So we have time for some quick